Picture yourself standing in front of a bathroom mirror before sunrise, half awake, toothbrush in hand, staring at the face that evolution left behind. The eyes, the jaw, the skin, the little patchwork of traits that make you recognizably human. Now imagine the mirror changes. It stops showing your face and starts showing the machinery underneath it. Cells stacked inside tissues, chromosomes folded inside nuclei, DNA packed into every living corner of you like a library so old that no one remembers who built the first shelf. And somewhere inside that library are empty spaces. Not blank pages waiting to be written. Not mistakes someone forgot to correct. Empty spaces where instructions used to be. Genes that your distant ancestors once carried, used, and passed down, until somewhere along the road to us, they broke, faded, or vanished from the working script. Which is a little unsettling, because we usually like to tell the story of evolution as if it were a long series of upgrades. Life crawls out of the water. Limbs get stronger. Eyes get sharper. Brains get bigger. Hands get cleverer. Eventually, after enough dramatic music and a respectable number of Ice Age close calls, here we are, making coffee, filing taxes, and using our oversized primate brains to wonder why our backs hurt so much. It feels like progress should mean addition. More tools. More abilities. More complexity. More of the good stuff. But the genome does not read like a clean success story. It reads more like an ancient crime scene. There are clues everywhere, but some of the most important ones are not the things still standing. They are the outlines of things that used to be there. A gene that works in other mammals but is damaged in us. A stretch of DNA that looks like an instruction manual with the key sentence crossed out. A molecular fossil, not preserved in stone, but buried inside the cells of living bodies. And the strange part is that these losses may not just be debris from evolution's junk drawer. Some of them may have helped shape what we became. That is the twist hiding in the human blueprint. Sometimes evolution builds by taking away. Not always on purpose, of course. Evolution is not an engineer in a white coat, carefully sketching a better ape. It is messier than that. Mutations happen. Environments change. Populations shrink, spread, collide, and survive by a combination of biology, chance, and timing that would make any detective slightly nervous. But when a gene is lost and the lineage keeps going, the loss itself becomes a clue. It raises a question. Why did that missing piece not destroy the organism? Did something else take over its job? Did the world change so that the gene was no longer useful? Or, more intriguingly, did losing it open a door that had been closed before? Because a genome is not just a list of parts, like the inventory of a hardware store. It is more like an orchestra, where turning one instrument down can change the whole song. A gene can influence when a tissue grows, how strongly a signal is sent, how a muscle forms, how a nerve develops, how a body responds to infection, food, stress, or injury. So when a gene stops working, the result is not always simple subtraction. Sometimes it is a rerouting. A switch flips. A brake releases. A pathway quiets down, and suddenly another part of the body has room to change. That is where this story gets interesting, and also kind of rude to our favorite myths about ourselves. We love to imagine humans as the deluxe edition of the primate line, the version with extra features installed. But what if some of the features that made us different came from things our ancestors lost? What if part of the path to being human was not a climb toward perfection, but a long, strange trail of trade-offs, broken instructions, and vanished abilities? Hidden inside your cells are traces of that trail. Not bones in a desert. Not footprints in volcanic ash. Something quieter. Older. A set of missing genetic lines that scientists can only reconstruct by comparing us to our living relatives and to other branches of life. And when those comparisons light up the places where our genome went silent, they do not just tell us what we lack. They begin to tell us what those losses may have made possible. So the question is not simply which genes did we lose along the way. The real question is much stranger. How much of what makes us human was shaped by the things evolution erased? Long before there were cities, campfires, stone tools, or anyone around to wonder what a gene was, The story was already being edited in the dark. Imagine a small primate moving through an ancient forest, its hands curled around branches slick with rain, the air thick with leaf rot, insects, and the warm animal smell of other bodies nearby. It does not know it is part of a lineage. It does not know that millions of years later, one strange branch of its family tree will stand upright, make music, bury its dead, launch machines into space, and still somehow lose its keys every morning. To that primate, survival is immediate. Find fruit. Avoid teeth. Keep close to the group. Get through the night. But inside every cell of that animal, something quieter is happening. DNA is being copied. Not perfectly, because biology is not that polite. Tiny changes slip in. A letter shifts. A sequence breaks. A gene that once sent a signal at the right time begins to stutter. Most of these changes vanish with the bodies that carry them. Some are harmful enough that they do not last. Some do almost nothing at all. But a few drift forward, generation after generation, like a smudged fingerprint passed through deep time. And that is where the comfortable version of evolution starts to wobble. Because when we picture the road from those ancient primates to us, we tend to imagine a highlight reel. The first steps on two legs. The hands becoming free. The skull changing shape. The brain expanding. The face flattening. The body losing much of its fur. The world becoming less like a forest ceiling and more like an open, dangerous, sunlit stage. It is a good story. It is dramatic. It even has decent lighting. But it makes evolution look cleaner than it was. It suggests that the path to being human was mostly a matter of gaining the right upgrades at the right time. And then you look inside the genome, and the evidence starts whispering something less tidy. There are places where our DNA seems to say, yes, something was here. A working instruction. A tool. A molecular machine. But now it is damaged, disabled, or gone from active use. Other animals still carry versions that work. Sometimes our close relatives do. Sometimes more distant mammals do. Sometimes the trail stretches so far back that it points to a feature our ancestors inherited from creatures that would never have looked remotely human. And yet, on our branch, the instruction went quiet. That is not the kind of clue you can put in a museum case next to a jawbone. It has no dramatic teeth, no claw marks, no skeleton curled in ancient mud. It is more like finding a locked room in a house you have lived in your whole life, then realizing the lock is older than your species. For scientists, these lost or broken genes can be unsettlingly useful. They are not just genetic clutter. They are timestamps, suspects, and sometimes alibis. If a gene works in many mammals but not in humans, that raises one kind of question. If it works in chimpanzees but not in us, that raises a louder one. If the same gene is broken in all living humans, then the break may have happened before our species spread across the planet. And if the loss lines up with changes in anatomy, diet, disease resistance, reproduction, or the way our bodies develop, then suddenly that missing instruction looks less like background noise and more like a clue left at the scene. But here is the part that makes the case harder. A gene can be lost for more than one reason. Maybe it stopped being useful because the environment changed. Maybe another gene took over its job. Maybe the loss was just tolerated, not selected for, drifting through a small population because chance sometimes has terrible handwriting. Or maybe, in a few important cases, losing the gene actually changed the rules. It removed a constraint. It made an old pathway weaker, or a new pathway stronger. It did not add a new feature like installing an app. It changed the operating conditions. And when the body is a network of signals, switches, brakes, and feedback loops, that kind of change can echo. Picture an ancient trail through a forest. One path collapses under a fallen tree, and suddenly every animal starts using a different route. At first, it looks like damage. Over time, it becomes the road. That may be how some genetic losses work. A door closes, and the lineage survives by moving through another one. But if that is true, then the human body is not only a record of successful inventions. It is also a map of closed doors. So when we look at ourselves and see the familiar signs of humanity, the big brain, the expressive face, the vulnerable skin, the strange mix of strength and fragility, we have to ask a more uncomfortable question. Are we looking at what evolution built, or at what was left behind after it took certain pieces away? The first real clues do not look like clues at all. They do not glitter in a drawer. They do not sit in a glass case with a little museum label. They are not fossil teeth pulled from a cave floor or footprints hardened in volcanic ash. They are made of letters, or at least the closest thing biology has to letters, lined up in long molecular sentences inside living cells. And to find them, scientists have to do something that sounds almost impossible. They have to look for evidence of something that is no longer working. Imagine opening several old family photo albums at the same time. In one album, a great-grandparent is standing beside a strange tool. In another, a cousin has the same tool. In another, a more distant relative has it too. But in your album, there is only an empty outline on the table, a pale rectangle where something clearly used to be. That is the kind of pattern that can make a genome start to feel haunted. When researchers compare human DNA with the DNA of other animals, they are not just looking for what makes us special in the obvious way. They are not only asking, what did we gain? They are also asking, what did everyone else keep that we somehow lost? And this is where the story becomes less like a straight road and more like a cold case. Take a stretch of DNA that works in chimpanzees, gorillas, orangutans, or other mammals. Maybe it helps build a protein. Maybe it helps regulate when another gene turns on. Maybe it plays some role in smell, muscle, immunity, digestion, development, or reproduction. Then compare it to the human version. Sometimes the human copy is still there, but it has a broken piece, like a snapped key stuck in an old lock. Sometimes the sequence is scarred by mutations that prevent it from making the same working product. Sometimes the gene is not exactly gone, but it has become quiet, like a machine unplugged in the corner of a room. And sometimes what remains is only enough to say, something used to happen here. That is a strange kind of evidence, because it asks scientists to treat absence as information. In paleontology, absence is always dangerous. A missing fossil does not always mean a missing animal. Maybe the rocks were destroyed. Maybe the body never fossilized. Maybe no one has dug in the right place yet. But in genetics, when the same missing or damaged instruction appears in every human population, and when relatives outside our lineage still have a working version, that absence begins to sharpen. It becomes less like a gap in the record and more like a mark on the timeline. Somewhere after our ancestors split from other apes, but before all living humans inherited the same genomic baggage, something changed. A molecular tool broke, and our lineage kept going. Which is already interesting. But the more unsettling question is not just how it broke. It is why the break survived. Because a broken gene should raise suspicion. If that gene was important, losing it should have been bad news. If it was not important, then why did so many other animals keep it? And if the loss was not just harmless, if it nudged the body in a new direction, then we are no longer looking at a random scratch in the genome. We are looking at a possible turning point. Of course, no single missing gene can explain humanity. That would be way too convenient, and biology loves ruining convenient stories. Humans did not emerge because one genetic switch flipped and suddenly an ape stood up, got philosophical, and invented airport security lines. The real story is tangled. Many genes changed. Many environments shifted. Many populations lived and died without leaving descendants. But that is exactly why lost genes matter. They are not magic buttons. They are pieces of a pattern. They can point to old pressures, old trade-offs, old bodies adapting to old worlds. They can show where a pathway became less useful, where a function was replaced, where an ancestral ability faded because the cost of keeping it no longer made sense. And sometimes they suggest something even weirder, that losing a function may have changed what other parts of the body were free to do. Picture a dark control room deep inside an ancient organism. Switches hum on the walls. Some control smell. Some control muscle. Some control cell growth, or immune defenses, or how tissues form before birth. Now imagine one switch breaks. The room does not explode. The body does not vanish. Instead, the signals reroute. Other switches take on more importance. A system that once had to work around that old signal begins to behave differently. Over thousands of generations, that difference can become part of the species itself. This is why the genome can feel less like a blueprint and more like an archaeological site. Every living human carries layers of ancient edits, and not all of them are additions. Some are erasures. Some are abandoned corridors. Some are doors that closed so long ago that we only recognize them because our relatives still have the keys. And now the case is starting to narrow. If scientists can identify these missing instructions, and if they can figure out when they disappeared, they can begin to ask what was happening to the body at the same time. Were our ancestors changing what they ate? How they moved? How they fought infection? How their brains and faces and muscles developed? The first clue is the absence itself. But absence is only the beginning. The real mystery is what changed after the silence began. The trail begins with a gene that once looked perfectly ordinary. Not dramatic. Not cinematic in the way a saber-toothed skull is cinematic. No giant tooth. No claw mark. No volcanic ash layer freezing a disaster in place. Just a small stretch of DNA, tucked inside the genome, doing what genes do for millions and millions of years, carrying instructions from one generation to the next. In many mammals, one such instruction helps make a protein involved in building a sugar molecule that sits on the surface of cells. That sugar has a very unglamorous name, and honestly it sounds like something you would rather not have to say out loud at dinner. But its job matters, because the surfaces of cells are not just smooth little balloons. They are covered with molecular tags, signals, and decorations that help the body interact with the world. They are part of how cells recognize each other, how microbes recognize hosts, and how immune systems decide what looks familiar and what looks suspicious. So far, that sounds like a normal piece of biology. Tiny, complicated, easy to ignore. But then scientists compared humans with our closest living relatives, and the normal story developed a crack. Chimpanzees and many other mammals still carry a working version of this gene. Humans do not. In us, the instruction is broken. A tiny deletion knocked it out, like a missing tooth in a gear. And because all living humans seem to share this broken version, the damage was not some recent accident in one unlucky family. It happened deep in our past, before modern humans spread across the world, and after our lineage had already split from the ancestors of chimpanzees. Which raises a question that is much bigger than one damaged gene. Why would a lineage survive after losing a cell surface molecule that so many other mammals kept? At first glance, that sounds like a loss, plain and simple. A useful molecular tag disappeared from our cells. Case closed. But biology is rarely that tidy. Because cell surfaces are also battlefields. Viruses, bacteria, and parasites do not float through life politely asking permission to infect. They grab onto molecules. They exploit doorways. They use the body's own labels as handles. So if a pathogen depends on a particular surface sugar to invade cells, then losing the gene that helps make that sugar could suddenly become less like breaking a tool and more like removing the doorknob before the burglar arrives. That is one possible suspect in the case: disease pressure. Maybe the loss spread because it made some ancestors more resistant to an ancient infection. Maybe it changed the way their immune systems recognized threats. Maybe it came with costs, but the cost was smaller than the danger it helped avoid. However, the clue does not stop there, because this missing molecule may not have affected only microbes. Cell surface sugars can influence many interactions inside the body, and changing them can ripple through biology in ways that are hard to predict. So now the mystery has two layers. The first is why the gene was lost. The second is what the loss did afterward. Imagine an ancient population of apes living in a world full of unseen enemies. Not lions, not leopards, not snakes in the grass, but microscopic things moving through blood, saliva, wounds, and shared spaces. No one in that population knows any of this is happening. There is no medicine, no microscope, no theory of infection. There is only survival. Some bodies are more vulnerable. Some are less so. A mutation breaks an instruction. Maybe it is harmful in one way, helpful in another. Maybe most days it barely matters at all. But then the environment changes, or a pathogen sweeps through, and suddenly the broken instruction is no longer just genetic noise. It is a difference between bodies. It is a clue written in who lives long enough to leave descendants. And then, over generations, that broken version spreads until every human alive inherits the silence. But here is where the story becomes even stranger. When scientists find a lost gene like this, they are not finding a single answer. They are finding a doorway into an older world. A world where our ancestors were not yet human, but were already becoming separated from other apes by invisible molecular choices. A world where a change too small to see could alter the surface of cells across an entire body. A world where the line between loss and adaptation might depend on what was trying to kill you. And that means the missing gene is not just a broken piece of DNA. It is a fossil of a conflict we cannot see directly. No bones. No battlefield. No preserved pathogen waiting in the rock. Just a scar in the genome, and a question wrapped around it. Did our ancestors lose something because they no longer needed it, because chance let it drift away, or because losing it helped them survive an enemy that has vanished from the scene? And if one missing instruction can point to an ancient war between body and world, then how many other silent places in our DNA are not empty at all, but evidence? The first missing instruction gives us a suspect, but not a verdict. And in this kind of case, that matters, because the genome is very good at making a mystery look simpler than it really is. Picture a detective bored in a dimly lit lab. One thread runs from a broken gene to ancient disease. Another runs to immunity. Another runs to chance. Another to the strange, ordinary messiness of reproduction and inheritance. At the center is that same uncomfortable clue from before, a molecular tool that many mammals still use, but that our lineage somehow lost. The tempting story is easy. A pathogen appeared. The old cell surface molecule became dangerous. A mutation removed it. The bodies carrying that mutation survived better. Mystery solved. Roll credits. But then scientists do the annoying thing that makes science actually work. They keep asking what else could explain the same evidence. Because a lost gene is not like a confession. It is more like a muddy footprint. It tells you someone was there, but not whether they were the culprit, the witness, or just a very unlucky bystander. One suspect is natural selection. Maybe losing that gene gave our ancestors an advantage, especially if some ancient microbe was using the missing molecule as a doorway into their cells. That would make the loss feel almost elegant, in a grim evolutionary way. Close the door, slow the invader, survive the outbreak. But another suspect is genetic drift, which is much less cinematic and much more chaotic. In a small population, a mutation can spread not because it is brilliant, but because the individuals carrying it happen to leave descendants. No grand strategy. No heroic adaptation. Just chance, doing its usual suspicious little shuffle in the background. And then there is a third suspect, trade-off. Maybe losing the gene helped in one context and hurt in another. That is often how evolution does business. It does not hand out clean upgrades like a video game. It negotiates. A change that protects against one disease might increase vulnerability to another. A tweak that changes the immune system might also affect fertility, development, inflammation, or how cells communicate. The body is not a set of isolated drawers. It is a crowded kitchen where every cook is bumping into every other cook, and somehow dinner still has to happen. So when one ingredient disappears, the whole recipe can shift. And here is the mini twist. The most interesting question may not be whether losing the gene was good or bad. It may be that those categories are too small. A genetic loss can be harmful, helpful, neutral, or all three depending on the environment, the moment in history, and the rest of the genome around it. That means the same missing instruction can look like damage from one angle and adaptation from another. Which is exactly why early interpretations of lost genes can be tricky. For a long time, broken genes were often easy to mentally file away as genomic leftovers, the dusty storage boxes of evolution. If a gene no longer worked, maybe it was just decay. Biology tried something, stopped using it, and the sequence slowly fell apart. And sometimes that is true. There are plenty of genetic remnants that look like old machinery rusting after the factory closed. But as researchers compared more genomes from more species, a different pattern became harder to ignore. Some losses are not random clutter. They appear at particular branches of the evolutionary tree. They line up with changes in bodies, behaviors, environments, or disease pressures. They show up not as meaningless scraps, but as events. As moments. As places where the story took a turn. Think of it like walking through an abandoned town. One broken window does not tell you much. But if every house on one street has the same door removed, and nearby towns still have those doors, suddenly you are not just looking at decay. You are looking at a pattern. Maybe people removed them for firewood. Maybe floods damaged only that block. Maybe some danger made doors a liability. You still do not know the answer, but you know the absence is organized. That is what makes lost genes so powerful as evidence. They force scientists to ask when the loss happened, who inherited it, what other species still have the working version, and what else was changing at the same time. But they also force a kind of humility, because genomes do not come with subtitles. A broken gene can tell us that something happened. It cannot, by itself, tell us the whole why. And the deeper scientists look, the more the case expands beyond one gene. Humans carry many genetic losses compared with other animals, and they do not all point to the same kind of story. Some may be tied to smell, which makes sense for a primate increasingly guided by vision and social cues. Some may connect to diet or digestion, as environments, foods, and eventually cooking changed what our bodies had to handle. Some may touch immunity, where the arms race with microbes can turn even tiny molecular differences into life-or-death stakes. Some may have shaped development in ways we are only beginning to understand. But each one comes with the same warning label: do not assume loss means failure. A missing gene is not automatically a weakness. It is a question. And sometimes, the question is not why did this break? Sometimes it is, what became possible after it broke? That is where the old upgrade myth really starts to collapse. Because if the genome is full of both inventions and erasures, then the human body is not a monument to constant improvement. It is a record of experiments, compromises, lucky accidents, and ancient pressures we can only reconstruct from scraps. The suspects are still on the board. Selection, drift, disease, trade-off, changing environments. Maybe several of them at once. And somewhere between them is the harder truth: the road to us was not paved only by what our ancestors gained. It was also shaped by what they could afford to lose. By now, the case has started to change shape. At first, a lost gene looked like a missing part. Then it became a clue. Then it became a suspect in a much larger investigation involving disease, chance, trade-offs, and the ancient environments our ancestors had to survive. But now the clues begin to connect in a more unsettling way, because genes do not disappear into empty space. They disappear inside bodies that are moving through real worlds. Worlds with predators, parasites, droughts, forests, grasslands, food shortages, injuries, births, migrations, and social lives complicated enough to make any primate wish for a quiet afternoon. So start with the missing gene as an instruction goes silent. Which raises the first question: what does a body do when an old instruction is no longer available? The easy answer would be that it simply loses a function. But the better answer is messier. The body reroutes. Other genes keep working. Development continues. Cells still divide, tissues still form, immune systems still patrol, muscles still contract, babies still have to be born. And if the loss does not end the lineage, then the body has already answered part of the mystery. It has found a way to keep going. That brings us to B, compensation. A lost gene may be cushioned by other genes doing similar jobs, or by biological pathways flexible enough to absorb the damage. Think of it like an old city after a bridge collapses. At first, the missing bridge is the story. Traffic stops. Routes break. The map looks wounded. But then people begin taking other streets. New habits form. Neighborhoods that were once quiet become important. Over time, the city is not just a city with one less bridge. It is a city reorganized around the absence. Which raises a second question. If the body can reorganize around a genetic loss, could that reorganization itself become evolutionary material? That is where the case gets stranger, because compensation is not always just repair. Sometimes it creates new pressures. If one pathway weakens, another may become more important. If one sense becomes less central, another may take on more weight. If a cell surface molecule disappears, immune defenses may shift around that absence. If a gene involved in anatomy or development stops working, the effects may not be dramatic in one generation, but across thousands of generations, small changes can become a different kind of body. So now we arrive at C, context. A gene loss does not mean the same thing in every world. In a dense forest, a change in smell might matter differently than it would on open ground. In a population facing a new pathogen, a change on the surface of cells could matter more than it did before. In a lineage experimenting with new diets, new social behaviors, new movement, or new developmental timing, an old instruction might become less essential, or even restrictive. The environment does not write the genome directly, but it does decide which edits can survive. And then, with context in place, we can turn back to A and see the missing gene differently. It is not just a broken sentence. It is a sentence broken in a particular body, at a particular moment, in a particular world. That is why a gene that looks like a loss in isolation can become part of a larger transformation when you place it back into deep time. And this is where anatomy enters the room like another witness. The human lineage was not changing only at the molecular level. Bodies were changing too. Teeth, jaws, brains, limbs, skin, guts, muscles, and immune systems were all caught in a long, uneven process of becoming something different from the apes we still share the planet with. Fossils give us pieces of that transformation in bone and stone. Genomes give us another set of pieces, written in code rather than mineral. And when those two lines of evidence begin to point in the same general direction, the story gains weight. Not certainty, because certainty is rare in a case this old. But weight. A skull can show that a face changed shape. A tooth can hint at diet. A pelvis can suggest a new way of moving. And a lost gene can ask whether some invisible molecular shift helped make those visible changes easier, harder, riskier, or possible at all. But here is the missing piece that keeps the loop open. Connecting a lost gene to a human trait is incredibly difficult. It is one thing to say this instruction broke in our lineage. It is another to say this break helped produce that feature. Biology does not like simple arrows. A gene can have more than one role. A trait can involve hundreds or thousands of genes. Environments can push the same body in different directions. Culture can change selection pressures. Fire can change food. Tools can change teeth. Social life can change survival. Disease can change who lives long enough to reproduce. So the evidence does not form a neat line. It forms a web. And in that web, lost genes are not the whole spider. They are strands that vibrate when something else moves. This is the acceleration point in the mystery. The old idea was that evolution mostly adds improvements. The first twist was that it also subtracts. The next twist is that subtraction does not stay subtraction. It can trigger rerouting, compensation, trade-offs, and new dependencies. A missing gene can become a pressure point inside the whole system. A closed door can change which hallway everyone uses. A silenced instruction can make another instruction louder. And after enough time, what began as absence may become part of identity. Which brings the case back to us. When you look at a human body, you are not seeing a clean list of upgrades. You are seeing a living compromise between old instructions, broken instructions, replacement routes, and environments that kept changing the rules. Somewhere in that compromise are the genes our ancestors lost. Some may be harmless debris. Some may be scars from ancient battles. Some may be the price of other adaptations. And some, maybe, help create space for traits we now think of as deeply human. But to see that clearly, we need one more kind of clue. Not just what disappeared. Not just when it disappeared. We need to know what changed after the disappearance, and whether the silence left a shape behind. And this is where the story turns on us a little. Because after following the missing genes, the broken instructions, the suspects on the board, it is tempting to imagine that somewhere in the genome, there must be a hidden switch for humanity. Lose this gene, gain a bigger brain. Break that instruction, free the hands. Silence this pathway, become the kind of animal that paints cave walls, tells stories, and argues with strangers on the internet. It would be a beautifully satisfying answer. It would also be almost certainly wrong. The big twist is that lost genes are not a secret shortcut to explaining us. They are not magic missing pieces that turn an ape into a human. They are something stranger, and maybe more important. They show that evolution is not mainly a story about becoming better. It is a story about becoming different, under pressure, with whatever parts are still available. That may sound like a small shift, but it changes the whole investigation. Because we tend to treat human traits as if they are trophies. Upright walking. Tool use. Language. Big brains. Long childhoods. Social learning. These are the shiny objects in the case file, the things that make us feel like the main character of life on Earth. But a genome full of losses makes that view feel a little too neat. It suggests that some of what we call human may have emerged not from a clean upward climb, but from a long series of trade-offs, constraints, accidents, and biological doors closing behind us. Picture a path through deep time, not as a staircase going up, but as a narrow canyon. At every turn, rocks fall behind the travelers. Some routes vanish. Some options are cut off. The group keeps moving, not because the path is perfect, but because it is the path that remains. After enough time, the canyon itself shapes the journey. That is the uncomfortable beauty of gene loss. Once an instruction disappears and a lineage survives, the body does not simply return to the old version. It adapts around the absence. Other pathways become more important. Other pressures take over. A missing piece becomes part of the architecture. And eventually, descendants may look at the final shape and mistake it for a design. But maybe some of that design is really the outline left by absence. This is especially unsettling when we apply it to ourselves, because humans are very good at turning survival into destiny. We look back and say, of course our ancestors walked upright. Of course their brains expanded. Of course their hands became tools for making tools. But at the time, none of it was guaranteed. The ancient world was not waiting for us. Forests shrank and expanded. Climate swung. Predators watched from grass and shadow. Diseases moved silently through bodies. Populations split, vanished, mixed, and scattered. In that world, a genetic loss might have been a liability in one environment and a lifeline in another. It might have mattered enormously for a few thousand years, then become background noise. It might have closed one possibility while nudging open another. So the old question, what did this loss do for us, may not be sharp enough. A better question is, what did this loss make the lineage work around? Because workarounds can become habits. Habits can become dependencies. Dependencies can become traits. And traits, after enough generations, can start to look like the whole point of the story. Think about the body as a city again. If a major road closes for a weekend, drivers complain. If it closes for a century, the city rebuilds around it. Businesses move. Neighborhoods change. New roads become main roads. People born later do not remember the old route at all. To them, the new map is normal. But hidden under that normal map is the history of a closure. Our bodies may carry that kind of history. A lost gene may have forced a rerouting so ancient that we no longer see it as rerouting. We just call it being human. And that flips the usual story inside out. Instead of asking only how humans gained extraordinary abilities, we have to ask what older abilities, signals, or constraints faded away as those new patterns took shape. Not because loss is always good. It is not. Many losses are harmful. Many are neutral. Some are probably just evolutionary clutter, the molecular equivalent of a broken appliance left in the garage because no one bothered to haul it away. But some losses may have changed the rules of the body in ways that selection could then work with. That is the key. Gene loss does not have to be the hero of the story to matter. It can be the missing bridge that changed the route. It can be the locked door that made another hallway important. It can be the silence that made another signal easier to hear. And once you see that, the human genome stops looking like a list of features we proudly acquired. It starts looking like a record of compromises. We are not the fully loaded model of primate evolution. We are one surviving draft, marked by edits, cuts, errors, repairs, and a few lucky revisions that happen to work well enough. Which means the traits we think of as our greatest advantages may be tied, somewhere in the background, to ancient losses we barely understand. That does not make the story smaller. It makes it bigger. Because now every missing gene asks the same eerie question, not just what vanished, but what did the vanishing make possible? And if we follow that question far enough, the line between strength and weakness starts to blur. A vulnerability may be the shadow of an adaptation. A limitation may be the cost of a different kind of survival. A broken instruction may be part of the reason another instruction mattered more. So by the time we reach the human body, with all its brilliance and absurd design flaws, the real twist is waiting there in plain sight. We may not be what evolution built by adding more and more. We may be what evolution shaped after taking certain things away. By this point, the investigation is no longer about one missing instruction, or even a handful of them. The clues have started pointing towards something bigger, a world where bodies, environments, diseases, diets, and genes were all changing at once, each one tugging on the others like strands in an ancient net. And that is exactly what makes the case so hard to solve. If you were following a fossil trail, you might hope for a dramatic layer in the rock, a single disaster horizon, a clean before and after. But the story of lost genes is not written that neatly. It is scattered across living genomes, ancient bones, changing climates, and the ecological pressures that kept rewriting the rules around our ancestors. Imagine standing on the edge of an African woodland millions of years ago, when the landscape was not one stable backdrop but a shifting mosaic. Forests expanded, then pulled back. Grasslands opened, then changed again. Rainfall patterns moved like a slow, planetary breath. The animals living there did not experience climate change as a chart. They experienced it as fruit that was harder to find, water that moved farther away, new predators in open spaces, new parasites in crowded groups, new foods that demanded different jaws, guts, and behaviors. And inside those bodies, the genome was not passively watching. It was carrying old tools into new situations. Some tools stayed useful. Some became less important. Some broke and barely mattered. And some losses, under the right pressure, may have become part of a new survival strategy. That is where the evidence begins to converge. Fossils show bodies changing, teeth, skulls, limbs, hands, faces, and pelvises shifting across the long and branching history of hominins. Archaeology shows behavior changing, stone tools, butchered bones, fire, migrations, and eventually symbolic traces that suggest minds interacting with the world in new ways. Ecology shows the stage changing, habitats opening and closing, food webs rearranging, disease pressures shifting as animals moved, grouped, scavenged, hunted, and shared space. And genetics adds a quieter but deeply weird witness, broken or missing instructions that mark places where our lineage stopped using something older. None of those witnesses can solve the case alone. A fossil jaw cannot tell you exactly which gene changed. A broken gene cannot, by itself, prove why a face became flatter or a diet became broader. A stone tool cannot explain an immune system shift. But when these different lines of evidence all point toward the same general idea, the outline becomes harder to ignore. Human evolution was not a clean upgrade sequence. It was a long negotiation with changing rules. And in that negotiation, loss was not just background noise. It may have been one of the ways the body kept renegotiating the deal. But here is the careful part, the part science insists on. Even when the mystery is getting good, not every lost gene has a dramatic meaning. Some are probably just remnants. Some may have drifted into silence without much consequence. Some may have mattered in ways we will never fully reconstruct. The genome is full of old rooms, and not every locked door hides treasure. However, the pattern still matters, because when losses cluster around functions like sensory perception, immunity, metabolism, reproduction, and development, they start to look less like random scratches and more like a record of changing priorities. A lineage that relies less on smell may tolerate the decay of some odor-related genes. A lineage exposed to new pathogens may benefit from changing the molecules on its cells. A lineage that changes diet, cooks food, shares resources, or moves through new habitats may find that some old biochemical tools are no longer as essential as they once were. And then the absence becomes historical. Not a hole in the story, but part of the story. This is the near solution hiding in the evidence. Evolution did not simply add humanity on top of an ape body. It edited an existing body under pressure from a changing planet. Climate altered habitats. Habitats altered food and movement. Food and movement altered anatomy. Social life altered survival. Disease altered the value of molecular defenses. And genes, gained or lost, helped determine which bodies could keep going through all of that. The Earth changed the rules, and the genome recorded some of the responses, not only in new features, but in abandoned ones. Which means those missing instructions are not isolated curiosities. They are small, silent pieces of a planetary story. A world gets drier here, wetter there. A forest opens. A population splits. A pathogen finds a doorway. A food source changes. A body adapts, or it does not. And somewhere in that chaos, an old gene goes quiet. The lineage survives. Other systems adjust. The absence stays. Millions of years later, scientists find the silence and ask what happened. That is why this case feels so close to breaking open. The answer is not that losing genes made us human in some simple, flashy way. The answer is stranger. The losses show us that becoming human was not just about acquiring new powers. It was about surviving a changing Earth with a body full of old instructions, some of which no longer fit the world around them. But one question still lingers, and it may be the most important one. If the planet helped decide which instructions our ancestors could afford to lose, then how many of the things we think of as uniquely human are actually the afterimage of worlds that no longer exist? So the answer, as far as the clues let us follow it, is not that humans were made by losing one perfect gene at one perfect moment. That would be too clean, and deep time is almost never clean. The better answer is that some of the things we lost became part of the conditions that shaped what we became. A broken instruction here, a silenced pathway there, a molecular doorway removed, an old sense weakened, a biochemical tool retired, and the body did not simply fall apart. It adjusted. Other systems took up more space. Other pressures became more important. Other traits had room to matter. And over enough generations, what began as absence could become architecture. That is the part that changes the way the human genome looks. It is not a pristine instruction manual for building the most advanced primate, which is probably good, because have you seen our knees? It is more like an ancient manuscript copied by hand through storms, famines, migrations, infections, bottlenecks, and accidents, with some passages added, some smudged, some crossed out, and some missing entirely. And yet the story kept being copied. Every person alive carries that strange manuscript. In your skin, in your blood, in the cells lining your gut, in the tissues that built your face before you were born, there are traces of decisions no one made. There are old solutions to extinct problems. There are scars from battles with microbes that may no longer exist. There are pathways that matter partly because other pathways went quiet. There are absences so ancient that we only notice them by looking sideways, at chimpanzees, gorillas, mice, dogs, fish, and all the other living libraries that kept different pages. That sideways glance is what makes modern biology so powerful. It lets scientists read the present as a set of overlapping histories. A fossil can show us a bone that changed. A stone tool can show us a behavior that mattered. A climate record can show us a world becoming colder, hotter, wetter, or drier. But a lost gene can show us something more intimate, a possibility that left the body and never returned. And when those clues line up, they reveal a version of evolution that is less like a ladder and more like a long, improvised survival story. Life does not simply climb toward perfection. It edits. It compromises. It doubles back. It loses things and then lives with the consequences. Sometimes those consequences are dangerous. Sometimes they are invisible. Sometimes they create vulnerabilities that never fully go away. But sometimes, in the right body, in the right population, in the right world, a loss can become part of a new way forward. That is not a moral lesson. Nature is not praising minimalism. Losing a gene is not automatically good, and gaining one is not automatically better. The point is stranger than that. Evolution works with whatever survives long enough to reproduce, and survival is always judged by the world at the time. On one ancient landscape, a missing molecular handle might protect against an infection. In another, it might do nothing. In another, it might come with a cost that descendants carry for millions of years. The same absence can be scar, shield, accident, and inheritance, depending on when you catch it in the story. And that means we have to stop imagining ourselves as the final polished version of our ancestors. We are not the completed edition. We are a draft that happened to make it through. A living arrangement of old gains and old losses, shaped by forests that vanished, climates that shifted, predators that watched from the grass, foods that changed, fires that burned, groups that moved, and microbes that pushed against ourselves from the inside of history. The missing genes did not single-handedly make us human. But they remind us that humanity was made through subtraction as well as addition. Through closed doors as well as open ones. Through silence as well as signal. And that may be the most mind-bending part. The body you are sitting in right now is not only a record of what evolution preserved. It is also a monument to things that are gone. Which leaves one last loop open. Gene loss did not stop with our ancestors. It is still happening, quietly, across populations, across species, across ecosystems responding to a planet that is changing again. Some losses will vanish without a trace. Some may become harmful. Some may be carried into futures no one is here to imagine yet. And millions of years from now, if some future scientist reads the genomes of our descendants, they may not recognize our world in our bones alone. They may have to look for the missing lines. They may find a silence in the DNA, and realize it was not empty. It was the shadow of a world that once existed, a world that shaped a body by taking something away.